Good afternoon, students. Now let us start biomolecule, which is a very important chapter in botany and also chemistry. And here, let us start the biomolecules. And in these biomolecules, now we may discuss about proteins and amino acids. So the first one, proteins. And what do you mean by proteins? And here, the Greek word proteos. Proteos means prime importance. That is prime importance. So, proteins are prime importance of the human being or living things. And proteins are biopolymers. Biopolymers of alpha amino acids. Alpha amino acids. Most of the proteins are made up of alpha amino acids. And these proteins may act as enzymes and hormone and antibodies. So here proteins on partial hydrolysis. Protein on partial hydrolysis. Partial hydrolysis gives peptides. And complete hydrolysis. Complete hydrolysis gives amino acids. Amino acids. Generally, these amino acids are alpha. Now, we may take amino acids. So, we are taking amino acids. Amino acids. So, protein on complete hydrolysis, it will be gives amino acids. Amino acids are the monomer of protein. So, it will be make a polypeptide chain and it gives the protein. Now, the amino acid itself, it is a bifunctional group. It is amino and acids. So, it is bifunctional group. One is amino. What is that amino group? It is NH2 group. Then it is the second acid group. Acid group C double bond O OH. So amino acid contains NH2 group, amino group and COOH group, acid groups. Okay. And here these amino group may be present as alpha or beta or gamma or delta. So we are taking here, that is R group and C, H and COOH. In an acid, the amino group is present on that alpha carbon. Then here are CH, NH2 and CH2, COOH. And it is alpha and it is beta. So here the amino group may be present in beta carbon or maybe that is gamma or delta. But all proteins is made up of alpha amino acids. So, proteins are made up of alpha amino acids. So, if we are taking the amino acids and the proteins, we are taking alpha amino acids. Alpha amino acids. And here all are in that L structure. It is in that L configuration naturally. And L means here it is COOH group, it is oxidizing group at the top, and reducing group R is on the bottom. And here it is NH2 groups is in L and hydrogen is left hand side. Then it will be reverse, then it is D configuration. So it is L and D configuration. Here it is COOH and R group, and here H and here NH2. So naturally occurring amino acids consist of alpha amino acid group and L configuration. Now, classification of amino acids. So it is classification. And there are two types of classification. The first one based on functional groups. So it is based on functional groups. Based on functional group. And it contains two functional groups. One is NH2 group and COOH group. So the first one, it contains one NH2 and one COOH. And you know that NH2 group is a base because of it contains lone pair. And COOH group is an acid. It is a proton donor. So that one NH2 group and one COOH group, then it is neutral amino acid. That is equal number of NH2 and COOH. Then second, it has one NH2 and two COOH group. Then it has one basic group and two acid group. Then the compound is acidic. Example, aspartic acid and glutamic acid. Aspartic acid and glutamic acid. Then the third one. Here it is 2 NH2 group and 1 COOH group. So the basic groups are higher, so that it is basic amino acid. And example, it is histidine, and arginine and lysine. These are all basic amino acids. Now, the second classification based on synthesis. Based on synthesis. There are 20 amino acids are there. Among these 20, 10 amino acids are prepared by our own body. By our own body. They are called as non-essential amino acids. Non-essential amino acids. And another 10 amino acids are given by a diet. It is given by diet. They are essential amino acids. Essential amino acids. Now, how will you remember these essential and non-essential amino acids? This is a shortcut here. Non-essential amino acids. It is prepared by our own body. Then it is the shortcut PVT, tin hall. PVT, tin hall. So that it is 10 amino acids are non-essential amino acid. The shortcut is PVT, tin hall. Then for essential amino acids, this also there is a shortcut. One is CATS triple GP. CATS triple GP. So here that is 5 and 5. So 10 essential amino acids and 10 non-essential amino acids. And each one we are taking each one. The first one, it is non-essential amino acids, that is PVT and tin hall. PVT, tin hall. What is it? PVT. It is phenylalanine. Phenylalanine. Then it is valine. Then it is tryptophan. Then it is threonine. Then it is isoleucine. And M for methionine. Methionine. And H for histidine. A for arginine. L for leucine. And it is lysine. And here these histidine, arginine, lysine are basic amino acids. Now these are all non-essential. Non-essential. It is PVT, tin hall. Now essential amino acids. Then it is CAS triple GP. It is C A T yes, CAS G P. Now it is cysteine. Then it is asparagine. Then it is aspartic acid. Then it is alanine. Then it is tyrosine. Then it is serine. Contains disulfide bonds. And it is glutamine. then glutamic acid, glutamic acid, then glycine and proline. So among these glutamic acid and aspartic acids are acid amino acids. That is, number of COOH group is greater than that of NH2 group. Okay. Now, the next one we are taking the properties of amino acids. And amino acids, I take here amino acids. Generally, it is alpha amino acid or CH, NH2 and C double bond O and OH. Now, it contains acid group and one basic group. What do you mean by acid? Acid is one, it is a proton donor. Proton donor. And a base is a one, it is proton acceptor. Now, it contains a bifunctional group. One is a proton donor and another one proton acceptor. So, acid donate a proton and a base, that is NH2 group, accepts a proton and make a sweeter ion. Here it is RCH and NH3 plus and COO minus. An amino acid containing two charges, which is known as Zwitter ion. It is called as Zwitter ion. Now, when Zwitter ion in that acid medium, here Zwitter ion is treated in acidic medium. Acidic medium. It is presence of H plus. Then it is at low pH. What will happen? It is acidic medium, so it will be get protonated so that the COO minus group is accepts one proton and become RCH NH3 plus and COOH. Correct? Now, it is a positive charged ion. Now, it is nothing but cation. It is a cation. So, at low pH, Zwitter ion converted into cation. Now, at high pH or in a basic medium, that is in presence of OH minus. At basic medium is nothing but at a high pH, that is pH is greater than 7 and here the pH is less than 7. At a high pH, in presence of OH minus, NH3 plus, that will be donate a proton and OH group accepts a proton. So this is the reacting one. Then it is undisturbed. So we are getting RCH, NH2 and COO minus, and it is a negative ion. The negative ion, which is known as anion. Okay. Now, a Zwitter ion in acidic medium may act as a cation, and Zwitter ion on basic medium it may act as an anion. Now, in a applied electric field, cation moves cathode, anion moves anode. So, when it is an applied electric field, in that electric field, cation will move towards a cathode. A cation will move towards cathode. And cathode is negative charged electrode. Now, It is an applied electric field. It is an anion. Anion moves anode. So it is moves on that anode, and anode is positive electrode. Now it is different pH. pH is less than 7 and pH is greater than 7, or acidic medium and a basic medium. So zwitter ion will move towards any of the electrode. Now at a particular pH, zwitter ion cannot move any of the electrode. There is no tendency movement of zwitter ion. So there is no movement at a particular pH. Then this is known as, or this particular pH point, which is known as isoelectric point. It is known as IP. So what is it? IP, isoelectric point. There is no tendency of a zwitter ion to migrate any of the electrode, which is known as isoelectric point. Now we are taking acidic and basic and neutral amino acids. So if it is acidic, then the pi is 3.0 to 3.2. Then if it is a neutral, then it is 4.6 to 6.5. Then if it is a basic, the isoelectric point 7.6 to 10.8. It is isoelectric point or particular pH. Okay. Now, what is the application of IP? Application of isoelectric point. At the isoelectric point, all amino acids are least soluble. These amino acids are least soluble. So this property is used to separate the individual amino acids. So it is easy to separate. So separation of amino acid is easier by knowing that isoelectric point. Now we may take another one property. Now we are taking an amino acid, that is glycine and alanine. So glycine, that is NH2 and CH2COOH, is nothing but glycine. It is a 3-letter symbol, and in glycine it is only one amino acid which is achiral. This is achiral. It does not contain asymmetric carbon, so that it is achiral. And when it is reacted with alanine, alanine is H2, N, CH and CH3 and COOH. It is alanine. Now it is a chiral carbon. It is asymmetric carbon. Now when glycine is reacted with the alanine, there are two possibilities. One is COOH group of glycine is reacted with NH2 group of alanine, so that it will make a peptide bond. It is H2, N, CH2 and C double bond O, and here NH and CH, CH3 and COOH. Now it is glycylalanine. In glycylalanine, COOH group of glycine is reacted and NH2 group of alanine is reacted, so that it will make peptide bond. It is a peptide bond. Now, one more possibilities are there. That is, COOH group of alanine is reacted with NH2 group of glycine. So that we are take alanine first. It is H2N CH CH3 and COOH, and it is alanine. Glycine H2N CH2 and COOH. Now, what is the reaction? Alanine and here it is glycine. Alanine COOH group, glycine NH2 group is reacted. Then we are getting that here H2N and CH, CH3 and C double bond O and NH and CH2 COOH. Now it is alanine glycine. So, two amino acids, one is glycine and alanine. It will be formed, that is, two peptide bonds. One is glycylalanine and alanine glycine. So that this peptide bond, which is known as, it is also a peptide bond. So, these two possibilities are possible, so that these peptide bonds are called as a dipeptide. So, what do you mean by dipeptide? Dipeptide means it is two amino acids are joined and form that a dipeptide. So that this peptide, which is known as dipeptide, it is also considered as a dipeptide bond. Now, a dipeptide cannot have two peptide bonds. It is only one peptide, is one peptide and another one. There is two possible is there, but it is never a dipeptide can only one peptide bonds. Same way, if you have taken a tripeptide which contains only two peptide bonds, but is the combination of three amino acids. So this tri and di represents number of amino acids undergoing the reaction. For any peptide, n-1 peptide bonds are possible. For any peptides, n-1 peptide bonds are possible. Okay, and the next one. The next one, we are take proteins. And here it is structures of protein. Structure of protein. Structure of protein is studied in 4 levels. So it is studied in 4 levels. 4 levels. One is primary and secondary and tertiary and quaternary structures. And each one level is somewhat complex than that of the previous. So, each level is complex structure than that of the previous one. Than that of the previous. Okay. Now we may take primary structure. Primary structure. Primary structure represents sequence of amino acids. It represents sequence of amino acids. And the sequence of amino acids that will be gives the biological function. So this sequence determine biological function. Biological function. Okay. For example, you know that hemoglobin. Hemoglobin. In hemoglobin, it is VHTPL. It is VHTPGL. And it is a sequence of valine, histidine, threonine, and proline, and glutamine, and lysine. And if one of the sequence, instead of glutamine, there will be a valine. Suppose here it is valine, histidine, tyrosine, and proline, and instead of glutamine, here there is a valine and lysine. Valine and lysine. And what is that? Here instead of glutamine, here there is a valine. Now it is not the hemoglobin here, it is sickle cell anemia. Sickle cell anemia. So if it is sickle cell anemia, the primary structure is valine. In this hemoglobin, the primary structure is that is glutamine. So if the sequence is changed, then the biological function is also changed.